So hello everybody, my name is Gergely Karsai. First, I would like to thank the organizer for the opportunity to be able to present my work here. And I would like to introduce our lab. So I'm working at the University of Zurich in the lab of Thorsten Bornemann. And we work on rare genetic diseases. Usually we get some samples from patients. We do lipid analysis. We come up with some interesting ideas that we would like to test later in cells, and then this circle repeats itself till the question is solved, if it's possible. So let's shortly to Swingolypids. I guess all of you already know what Swingolypids are. They are aliphatic amino alcohols, and the basic structure of the Swingolipid starts with the sphingolipid backbone, here represented in blue as the sphingosine, which can be either head group modified or N-acylated with various fatty acids, which forms then ceramide. Ceramides can be further head group modified to form sphingomyelin, for instance, or various glucosyl-ceramides. So these are the higher level Swingolipids. Swingolipids were long considered only a structural component of the cell membrane, as you can see here on the right, where they were highly enriched in lipid rafts. And as we already know, Swingolipids, mostly sphingomyelin, go hand in hand with cholesterol. But recently they function in somehow. I cannot move. Oh, no. So in the last 2 decades, they have been increasingly recognized as important signaling molecules, where sphingosine and ceramide have important signaling function. They tilt the cells towards apoptosis, whereas sphingosine 1-phosphate does pretty much the opposite. If we look at more complex organisms, they have usually 2 or 3 sources of Swingolipids, namely the ectopic uptake of Swingolipids or the endogenous de novo production of Swingolipids. In certain organisms, microbiota can play a big role, not only by supplying certain Swingolipids, but they can also influence the endogenous production of the organism. If we look in the human plasma and the Swingolipid backbone profile, we can see that a small percentage represents the so-called other Swingolipids. So these are the atypical Swingolipids, such as the deoxyswingolipids, which our lab is probably known for. And recently, Musilone from our lab published another variety of atypical Swingolipids in this nice publication. But for today's presentation, I would like to focus on the D18-based Swingolipid. So the D stands for the dihydroxy, and the 18 for the number of carbons in the backbone. And this 18 carbon comes from the conjugation of palmitoylclay with L-serine. And then the second number represents the number of desaturation of the Swingolipid backbone. So we can see that the double bond is introduced by an enzyme. It's either DEGS1 or DEGS2, which introduces a double bond at the delta 4 position. And later on, FAX3 enzyme can introduce a second double bond to form the D18 2-based Swingolipids. And as you can already see, DEGS1 has probably a very important function to establish this ratio in the plasma. And looking in the literature, there are several correlations to certain type of Swingolipids. So the D18-0-based Swingolipids, or other name for them, the dihydroSLs, were long considered as an early predictor of type 2 diabetes. But thanks to Thomas Hanif, it turns out that they were actually the deoxyswingolipids. But what we know mostly is the D18-1 Swingolipids, mostly the ceramides, which have a very bad reputation. They are not only predictor of type 2 diabetes and cardiovascular disease, but they can directly induce hepatic steatosis, insulin resistance, and beta cell failure. So they are relatively bad. On the other hand, the downstream metabolite of D18-1s They are inversely correlating with BMI, human index, type 2 diabetes, and cardiovascular events. So it would be very good if we could reduce the amount of D18-1-based Swingolipids, especially ceramides, by increasing the amount of precursors or the downstream metabolite. And Scott Summersell in particular was interested in this, and we can see that there are several animal models, not also from Scott Summers' group, the mice, but also fruit flies, and all of these animal models lack the DES1 enzyme, which results in the accumulation of the precursor and reduced product. This has positive and negative side effects. Negative side effects are neuronal generation and growth retardation in both, and the heterozygous mice, the DES1 heterozygous mice, had also improved glucose tolerance. So now there is a new mice model. It's a conditional DES1 knockout mice, which does not display any negative side effects, just the positive side effects of DES1 deficiency. Also, the pharma industry got very much interested in DES1 inhibitors because of its positive side effects. Unfortunately, we have also a story to tell, which starts with this patient who had early developmental retardation, physical as well as mental. He has spasticity in the extremities, epilepsy, cerebral atrophy, and hypomyelination, as you can see from the MRI of the patient. You can see here a reduced myelination in the cerebellum very nicely compared to the healthy volunteer. Whole exome sequencing of the index family revealed that our index patient has a homozygous mutation in DES1, whereas the unaffected siblings, as well as the parents, are heterozygous carriers. So DES1 is the Swingolipid delta 4 desaturase, and this mutation is at the evolutionary highly conserved region. We did the Swingolipid analysis of the plasma of the index family and also healthy volunteers, and we could detect that the patient has highly elevated dihydroSLs levels compared to the heterozygous parents or healthy control. The mutant DES1 localization was similar to the wild type localization. However, the amount of DES1 protein in patient fibroblasts was strongly reduced. This suggests that this mutation renders the enzyme less stable. Next, we tested the de novo synthesis in cells, and we could detect that in the patient-derived fibroblasts, the DES1 activity was reduced, which is represented here by the higher levels of dihydroSLs compared to the control plasma. But it was not completely abolished or like in the DES1 knockout cells. In addition, we also did long chain based analysis of this plasma, and we discovered an additional sphingosine isomer in the patient plasma, which was not present in the parent's plasma, nor in any controls that we tested. Further, we discovered this additional lipid was also present in DES1 knockout Hepburn cells, or if we pharmacologically inhibited DES1 by 4-HPR, we could also detect the appearance of this lipid. Further analysis revealed that this sphingosine isomer bears the delta 14C double bond instead of the canonical delta 4E double bond. So to summarize the first part, normally DES1 will be the first enzyme to desaturate the sphingolipid backbone to form the D18-1 base sphingolipids, and then FATS3 is making the second double bond at the delta 14C position. However, if DES1 activity is reduced, then we see that the precursor is accumulating, and then FATS3 can take over the desaturation as well. So we will have two sphingolipid backbone with different double bond position, one with the delta 14 and one at the delta 4. So this results in a distorted Swingolypid profile and also hypomyelination. Also, you could say that this is only one case, so it's not a tendency. But since then, two independent groups showed similar clinical symptoms in 19 and also 4 other patients from 14 different families, which all had DEGS1 mutation, which resulted in increased dihydroSLs accumulation and hypomyelination. So now we wanted to answer the question, which one of these metabolites, the increase or the lack of the metabolites, is causing the hypomyelination. And in order to do so, we wanted to move in vitro, so using cell culture, where we could either pharmacologically, genetically, or just saturate DEGS1 activity. And further, we wanted to obviously rescue this phenotype. So we do the following assay. So this assay is established at the University of Glasgow in the lab of Julia Edgar, where I could visit her in FAB's short-term fellowship. And we used mouse embryos. We dissected the spinal cord, isolated the neuronal precursor cells, and we did general differentiation regimen. And then this was followed by 2 weeks of different treatments. So 3 times a week, the treatment was exchanged. And after the end of the treatment, we harvested the cells either for MS, genetic analysis, or for immunohistochemistry. And for immunohistochemistry, we were interested in the degree of myelination. So we stained these slides with MVP, so this is myelin-basic protein, and an axon marker. And to calculate the myelination percentage, we applied cell profiler algorithm, where we quantified the amount of myelin-shaped structure which is covering the axon staining. So this will be later represented as percentage of myelination. So first we tried increasing concentration of 4-HPR, and what you can see, this is the quantification of the pictures we took, and you can see that axon density represented in red was not affected by 4-HPR treatment, but the myelination was strongly reduced. Later, we analyzed the Swingolypid profile of these co-cultures, and we could see dose-dependent increase in dihydroswingomyelins. But this already pretty much represented what we saw in the humans. We also tested another potent inhibitor of DEGS1, GT11. And with GT11, we also saw that the axon density was not affected, whereas we saw a slight decrease in the myelination. When we checked the Swingolypid profile of this culture, we noticed that the accumulation of dihydroswingolipids was much lower than in the case of 4-HPR. And this we also confirmed in HEC293 cells, and we also found that at higher GT11 concentrations, the de novo Swingolipid synthesis is inhibited. So probably this can explain why we don't see dose-dependent increase in dihydroswingolipids. So next, we came up with an idea which originated from this very nice publication, where the authors disinhibited SPT activity or overexpressed hyperactive SPT fusion protein, which resulted in the accumulation of various Swingolipid species and also demyelination. So with this in mind, we supplemented the free base swingonin to the cells, and then we were hoping to have highly increased dihydroswingolipid levels in this culture. So this is the percentage of D18-0 based Swingolipids in these cultures. And unfortunately, the increase was not very high. And to confirm the phenotype, we also supplemented sphingosine, and there we observed the opposite. Unfortunately, this treatment had no effect on myelination. So next we move to the genetic approach. So we used siRNA to knock down DEGS1. Again, axon density was not affected. Myelination was significantly reduced. Then qPCR confirmed the knockdown of DEGS1. And Swingolipid analysis revealed that this also resulted in increased dihydroSLs levels. So next, we wanted to check whether is this the accumulation of dihydroSLs which causing the demyelination, or the lack of the product, the D18-1 based Swingolipid. So we treated cells with myriocin, and we saw basically no significant difference in axon density nor myelination, although the amount of Swingolipids was strongly reduced in these cultures. So this suggests that it's either the accumulation of the precursor, or it is this other sphingosine isomer which is produced by FATS3. So we also wanted to check whether this byproduct can be found in this culture. So the byproduct, so the sphingosine isomer comes here in the DEGS1 knockout cells with labeled after treatment with deuterium labeled swingamine. You can see this is the unlabeled form, much less. But increasing concentrations of 4-HPR in this culture did not result in the appearance of this pre-peak. And this can be explained by the low expression of FATS3 enzyme in the mouse brain. So this is data from this brain RNA-seq.org homepage. And also our collaborator had mRNA-seq data from mice, and FATS3 was not amplified. So next, we wanted to inhibit the formation. So while we block DEGS1 activity pharmacologically, we also wanted to inhibit the formation of the precursor. So therefore, we used either myriocin or FB1. So this is just the Swingolipid profile of HEX cells, either treated with myriocin, FB1, or 4-HPR and the combination of FB1 and myriocin. And we can associate that 4-HPR increases the formation for the amount of dihydroSLs. FB1 has basically no effect on this, and myriocin will rescue the Swingolipid profile. So we wanted to test this in neural co-culture system. So we checked again myriocin alone, HPR, high increase in dihydroSLs levels in the combination of 4-HPR with myriocin, rescued the Swingolipid profile. However, the myelination was not improved. So we repeated this experiment many times with different concentration. There was no improvement in myelination. So we looked back in literature, and it turns out that 4-HPR itself increases ROS production. And if we add myriocin as well, so here we can see that 4-HPR in two different cell lines increases the precursor, the dihydroceramides. In combination with myriocin, this increase is abolished. And then when the authors checked the ROS production, they saw that 4-HPR alone, as well as in combination with myriocin, produces basically the same amount of reactive oxygen species. So this would suggest that in this case, where we could not rescue the 4-HPR treatment with myriocin, is due to the ROS effect. And this is also supported by previous publications. So This is our data where we just tested in the same cells that the synthetic Swingolypid dihydroceramide increases ROS as well as hydrogen peroxide, and pointed out showed that the DEGS1 mutant fibroblast, so this is patient-derived fibroblast which bears DEGS1 mutation, they have increased ROS levels and also hypomyelination. So probably there is a link to ROS. And also there is a preprint out already that claims that if DEGS1 activity is reduced, then this not DH oxidase complex gets constantly activated by REC1, and this produces high amounts of reactive oxygen species, which they showed that it also affected neuronal growth. So with this, I would like to summarize what we found, that DEGS1 inhibition in vitro decreased myelination in a dose-dependent manner, whereas it had no effect on axon density at all. And GT11 was less potent than 4-HBr, probably due to the inhibition of the sp3 complex. And we could confirm our pharmacological inhibition by genetic ablation of DEGS1. And surprisingly, this delta 14 swingers in isomer that we found in the human samples was not found in the mice, probably due to the low Fas3 expression in this culture. And unfortunately, miriasin did not rescue the 4-HBr caused myelin effect. However, it restored the accumulation of dihydroSLs. And I would like to thank our lab, and also especially Dr. Julie Edgard, who received me in her lab, and also Shazia for helping with the experiments and also partially taking on this project. And also I would like to thank our German collaborators, partner with whom we published the first DEGS1 paper from our side. And I would like to also thank for the FABs for sponsoring my visit in Glasgow, and also for your attention.